Welcome everyone to the first of this webinar series on Introduction to Designing Adaptive Trials Using Facts, and to today's topic, which is doing dose escalation designs. This is in webinar mode, so everyone will be muted. If you have questions or comments, do please use the Q&A or the chat, and my colleague Code Feely will keep an eye on the traffic and in those and will let me know if there's a question that needs addressing. So what we're going to do is look at an introduction to the problem of designing a dose escalation trial. Then we'll have a worked example looking at designing a trial using facts, using I3+3, using BOIN. And then towards the end, we'll consider what the implications of FDA's Project Optimus is for designing dose escalation trials, where they're stressing the need to better identify the dose for phase 3. We'll have a quick look then at what we can already do in facts to target efficacy in dose escalation. And then at the very end, we'll have a quick look at what we hope to be doing in dose escalation in facts in the next year or so. So the dose escalation problem. As I'm sure you're all well aware, dose escalation is typically done in small cohorts, maybe 3 at a time is the convention. Each cohort's results are collected, and once they're available, the decision is made as to what the next cohort should be given. And in many cases, this choice is limited to escalating to the next dose, staying at the same dose, or de-escalating to the previous dose. Other designs can allow bigger jumps up or down, but most of the models have just those 3 options. And the important thing to consider in dose escalation is just how little information we're basing our decisions on. So for instance, if we've treated a cohort of 3, we might see 0, 1, 2, or 3 toxicities after we've waited the allotted time and have to make a decision based on that. And here in this graph, you can see the resulting beta distributions from having seen 0, 1, 2, or 3 toxicities. And so, and these represent what our probability distribution is of what the toxicity rate is on the dose that's just been tested here on the x-axis. So you can see if we've seen 1 toxicity, not unnaturally, the peak is around about 0.3, but there's a huge possibility that the toxicity is below that or above 0.3, all the way up to 0.8 or more. Similarly, if we've seen 2 toxicities, although it now becomes a lot less likely that the toxicity rate is 0.3, there is still some possibility. And really, it's only if we've seen 3 toxicities that we're pretty darn sure that the toxicity rate on this dose is above 0.3. Sadly, even after we've tested more subjects, say a second cohort, we've still got quite a lot of uncertainty. So again, now looking at these 7 distributions with the 7 different possible numbers of toxicities that we see from a cohort of 6. Again, if we're looking at for a toxicity rate of 0.3 or 0.25, there's several outcomes which still leave us with that possibility. So no toxicities, 1 toxicity, 2 toxicities, 3 toxicities, all give us still a good probability that our toxicity is in the target band, but of course no guarantee that it is. And really, it's only with 5 or 6 toxicities that we're really sure that we're above the band. And there is no result that makes us sure that we're below the band. And so on and up to 12 subjects. Each distribution is still quite is still quite diffuse. So at what is often the maximum or even above the maximum amount of data that we collect on a single dose in dose escalation, we've still got quite a considerable uncertainty about the true toxicity rate on the dose. And it's really important to hold that in mind. Flipping it around, if we knew what the true toxicity rate was, what would be the probability of seeing 0, 1, 2, or 3 toxicities? So this is just another way of trying to impress you with the uncertainty. So for instance, if the true toxicity rate is 10%, so below our, typically below the sort of rate that we're looking for in a dose escalation study, there's still a 25% chance or more that we'll see 1 or 2 toxicities out of 3. If the true toxicity is one of our target toxicities, say 0.3 or 0.25, then we've got over a 20% chance of, if the rate is 0.3, and over a 20% chance that we'll see 2 or more toxicities. So something that would normally cause us, that would typically cause us to de-escalate. And if the toxicity is well above what we're going to want, say 0.6, then we've still got some over 35% chance probability of seeing no or 1 toxicities, so of it looking benign. So again, just to reinforce that we mustn't overinterpret the number of toxicities we see at any one dose. So as a statistician, what are your first questions going to be to the team asking you to design a dose escalation study? So of course, the key first question is, what is this target toxicity rate? And of course, what are these toxicities that we're counting towards it? What's the target band for the toxicity? It's increasingly accepted that we shouldn't think of targeting a specific rate, given all this uncertainty that we can see in that we'll have after a limited amount of data. So it's much better to present some kind of upper and lower band around that target. And then what's an unacceptable probability that a dose has a toxicity rate above that target band? So if we're projecting toxicity rates at higher doses, what would be a projected probability of that the toxicity is above the target rate that would make us say, no, we're not going to escalate to that dose? Another crucial question, of course, is how many doses are there available to test? And for kind of operational aspects and thinking about sort of trade-offs of getting more data and but time to run the trial, how long do we expect it's going to take us to recruit a cohort? And then how long will we have to observe the cohort after we've treated them? For instance, if that observation time is very long, we might want to use larger cohorts, larger than 3, to try to get the trial done in fewer steps. It's surprisingly often that the team will actually have a very small number of doses they want to test, perhaps as few as 4, almost too few to be bothering with the design at all. And it's worth challenging them with questions such as, well, if we got through all 4 doses and see no toxicities, what would you want to do? And basically, they've got 2 choices. They can be so confident or that they don't want to go higher that they would say, well, that's it, we'll accept the top dose as the target, or they might say, oh, well, we'll want to go higher. And so we've extended our dose range now from 4 to 5 by adding a higher dose. And similarly, if at the very first dose we saw, say, 2 or even 3 toxicities, are they sufficiently convinced? Perhaps better ask this in the context of seeing 2 toxicities. Are they sufficiently convinced that this first dose is actually not very toxic, that they would be happy to redose at that level? Or actually, would they want to step down? So quite often they'll say, no, we prefer to step down. And actually, in practice, we can extend these initial 4 doses to 6 within a sort of a safety dose at the bottom, which we maybe don't start at. We maybe start at the second dose and a top dose, an extra top dose, in case we see very little toxicity. The other question about the doses is to ensure that they're confident that the spacing between the doses is correct. So say you're targeting a toxicity rate of 0.25, and maybe what we've set up as our target toxicity band a range from 0.16 to 0.33. These are kind of typical examples in the literature. The implication of that is that you'd really want the doses spaced so that if you've got a dose with a rate that's below the target, then the next dose shouldn't have a toxicity rate that's above the target, so that you miss the band altogether. So the doses ought to be in some way sufficiently closely spaced that we don't miss our target band. So if for convenience we assume that we're going to, in our model, so we're going to think about this in terms not of the things like the BOIN and MTPI for a moment, but the Bayesian Logistic Regression, where we fit a simple linear logistic curve. Assume that we're just going to have equally spaced dose. Assume our doses are equally spaced in terms of a potential toxicity, and we'll simply give them effective dose strengths of 0, 1, 2, 3, 4, etc. So what we'd like to be able to say is that if there is a dose I, such as that PI, our estimated toxicity rate, is below 0.16, then the maximum slope of our curve is such that the toxicity of the next dose, PI plus 1, should be such that PI plus 1 is less than 0.33. And so if we look at the difference in the logits, that means for 1 dose increment, we want the slope to have a value of no more than about 0.95. Call it around 1. If our expat values are not 1, 2, but something else, say we're doing some other log the dose transform, then of course you need to rescale the beta. So if we say that the maximum slope, we've asked the team to ensure that the doses are sufficiently closely spaced, that the maximum slope we would see is 1 in the log odds, then that might mean that if we sort of pick, say, a dose 4 as our target as an example, then if we then look at changing, so that gives us a log odds there of our target being 0.25, gives us a log odds of just below -1. Then if we look at us changing the log odds of 1 below that and above that, that would give an kind of expected scenario that would look like this, giving us a starting dose with an expected toxicity rate of about 0.05 and an emergency dose below it with a toxicity rate of about 0.016. And here you would hope, of course, during the dose escalation, that you wouldn't escalate much beyond dose 5, certainly not reaching dose 7. So maybe in this scenario, a dose 7 would be unnecessary. And then we might also think, well, what happens if we're really unlucky and the way the doses are spaced, it just so happens that they nothing falls right in the middle of the target band. Maybe we have a dose either side of it. Then we might imagine the scenario looking like this. But this ought to be with our maximum slope. What about the expected slope, or perhaps a possible slope below what's expected? So if our maximum slope has got a change in the log odds of 1, maybe the expected slope is say 0.66, and maybe the lowest slope, the shallowest slope we expect is maybe a log odds change of 0.33. These might give us scenarios that look like this. So again, with our starting doses at about a log odds of -3, so having a rate of about 0.05, we can see that at the lowest slope, we actually do need to go up to dose 8 to reach our target. Although dose 7 actually also lies within our target band, it's just not in the middle of it. And our median case would be that we'd get to our target toxicity at around about dose 5. Dose 6 is only just above the target. We'd quite often expect it to go to dose 6 and maybe even dose 7 before de-escalating to dose 6. So again, looking at things through this lens, hopefully we can see that even 6 doses may not be enough. We might need to go to 7 or 8 doses in case the toxicity doesn't increase the way it's expected. So part of the art of designing adaptive trials is the selection of the scenarios to simulate, and particularly in dose escalation, the scenarios we simulate are going to drive some of our design decisions. We're going to compare different options and how well they perform in the scenarios we've chosen as against other designs. So we want to be careful that our chosen scenarios don't have some kind of bias that favor a particular kind of design, and that they've got enough, they encompass our uncertainty sufficiently. At the same time, of course, we don't want to have too many scenarios, so many scenarios that we just end up in with a massive number of operating characteristics from a massive number of scenarios to compare, and it ends up being really hard to differentiate between designs because one design does better in one place and another in another, and so on. So almost a minimum, you could almost imagine a minimum set of scenarios by saying, well, in this scenario, we might misidentify the strength of the starting dose, a bit like we do in this example here, where we've got a log odds difference of about 0.03. We might have the difference we've looked at here, which of course is the a difference in slope. And we've also assumed that the slope is constant, and of course that may not be the case. Maybe the PK/PD doesn't work out quite the way the team forecast, and the spacing of the doses leads to either a decrease in slope between doses or maybe an increase in slope between doses. So with kind of 3 dimensions of uncertainty, we could easily yield at a minimum 3 by 3 by 3, 27 scenarios, which is already probably too many to contemplate. So maybe a central core would be, well, obviously we'll have a scenario with the median expected starting toxicity at the second dose, the median expected slope, and that the slope is constant. And then we could create 6 scenarios off of that. The first 2 where we change the expected starting expected toxicity at the expected starting dose, a couple where we change the slope, and a couple where we vary the median slope and make it increasing or decreasing. So as I said, choosing these scenarios to simulate this is, and we'll see this, I think, throughout the webinars, is a key part of the art of doing adaptive design. It's really important not to ignore any uncertainty and unintentionally build in some bias or expectation in the design, which may not be met when in the actual trial. But equally, it's important not to get bogged down with too many scenarios. And of course, if you have multiple dimensions of uncertainty, then of course, many of those different combinations of scenario combining different values on the different dimensions will be combining values, extreme values from your uncertainty. Therefore, they might be extremely unlikely. As a friend of mine memorably said, the ratio of the unit sphere, the volume of the unit sphere to the volume of the unit cube reduces drastically the higher the number of dimensions. It was memorable because it took me a while to work out what he just said. So one of the things, so as well as eliminating scenarios where the combination of extreme values means that particular scenario seems particularly unlikely, we can remove those. We may also remove scenarios where actually the simulation results are very similar, and we can concentrate on some smaller set of exemplar scenarios. Maybe moving back to right at the very end to a broader range, just as a sensitivity check. So that's the doses being included and the scenarios to be simulated. What about our sample size, our escalation, and our stopping rules? Okay, the first rule of thumb is you're clearly going to need as many cohorts as doses you're testing in order to be able to get to the top dose. You also need additional cohorts on top of that to reach the maximum number of cohorts you want on the selected dose at the very end. Plus, you may need another couple of cohorts in case on the way up to the top, a toxicity is seen and a second cohort is required on that dose before it's clear we can escalate beyond it. Or it may be you get to the top dose and then have to backtrack and allocate to a lower dose that you already tested on the way up. The allocation rules is very typical to require one cohort per dose and to only go up one dose increment. Perhaps that's a bit of a hangover from the original 3+3, but it's also baked into an awful lot of these newer methods that are improvements on the 3+3, but they're very much kind of decision-making methods rather than modeling methods, like the MTPI and BOIN that we'll look at in a minute. If you have a trial where, in fact, there's more than the sort of number of doses we've been talking about, that intermediate doses have been included, then maybe the escalation would be 2 of these dose strengths. So going up, you skip the half doses. That might be modified once you reach higher doses or perhaps after seeing a first toxicity. Another possibility is to include a run-in using smaller cohorts of just 1 or 2 subjects, which you start at the lowest dose and you go up until you see the first DLT. And probably that should be seeing a moderate toxicity, sorry, not a DLT, a toxicity, and that should be a moderate toxicity, not a full DLT. And then lastly, the stopping rules. Typically, particularly for the non-model designs, this is just in the number of subjects you tested on the doses being selected, and I think commonly that's now at least 9 subjects, perhaps more. And in the Bayesian Logistic Regression Method, we call N-CRM, in fact, there's a couple of additional rules that you can use. One is to use the a posterior probability that the current dose has a toxicity in the target band, where you might set a threshold of, say, 0.5 or 0.6. Setting one much higher than that, you can require much larger numbers on the dose. Or another possible rule is stopping at a dose if another cohort with no toxicities would not cause you to escalate, or of course, very unlikely to cause de-escalation, not cause escalation beyond the current dose. And then one way to recheck the sample size is to actually run the simulations with a stopping rule, and with a very large maximum, which you then expect not to reach, and then review the individual simulations, say 1,000, to see the actual largest sample size required. So before we try to expand that into an actual example, are there any questions, Kurt?
We have one, and it actually is, we have two now. First is, will the slides be available after the meeting? And certainly the recording will be. Tom, certainly the slides will be, but how people can get them. And the second is, what is facts? And I think that answer is forthcoming.
Right, yes. Yes, so the slides will be available. Obviously this, we will almost automatically send out a link to the recording for everyone who registered. I'll have to find out how we easily make the slides downloadable as well.
I assume we can host those on our website and put a link to that as well, I assume.
Yeah,
somehow.
yeah, I think so.
So,
so yes, what is facts? So facts is a general, is our software package for simulating fixed and adaptive trials, the fixed and adaptive clinical trial simulator. And within it, it has modules for doing different broad classes of trial, one of which is dose escalation, and it'll be those that we're looking at today. So, so this example was created in order to compare and contrast sort of three of the leading non-modeling approaches currently: the I3+3, the Bayesian Optimal Interval design, and MTPI, which I can't remember what that stands for. These are three really, as we'll see, really very similar approaches, although interestingly they have slightly different operating characteristics. And then what we call the N-CRM, others call the BLRM, the Bayesian Logistic Regression. The CRM is an old design in the 1990s, the continuous reassessment method. We appended an N to it, where N might stand for Neuenschwander, New, or Novartis, based on a 2008 paper by Neuenschwander from Novartis, where he addressed many of the, if not all, the shortcomings of the original CRM and made it into a much more practical, much more attractive method. So what I've attempted to do here is pre-specify an example test case. I haven't gone back and tweaked features of the test in order to favor one design or another, or even to just try to get a clear result. Coming into it, my expectations were that the I3+3, the BOIN, and the MTPI would be similar, and that in trials with small numbers of doses, the N-CRM would be similar too, but then in trials with larger number of doses, the N-CRM would be superior, particularly when the simulated MTD is at a high dose. And this is because what distinguishes the CRM methods is that because it's fitting a model through all the data points, it's borrowing information from the earlier doses onto the current dose in a way that the I3+3, the BOIN, and the MTPI don't. Now, at small numbers of doses, if the MTD is at the second or third dose, that really doesn't make much difference. But if the my expectation was if the MTD was at kind of dose 7, dose 8, somewhere out there, then this this information from the earlier doses would be useful. So the I3+3, the MTPI, and BOIN. So currently only one of these is in fact the MTPI. So I've been using, to simulate the I3+3 and the BOIN, I've been using packages that are freely available on the web. They're all very similar. They just look at the data on the current dose and they determine from that whether the next cohort should escalate to the next dose, stay at the same dose, or de-escalate to the previous dose. So for instance, the I3+3 simply looks at the ratio of the number of toxicities observed at the dose divided by the number of subjects tested. So that gives you two ratios, X over D and X minus 1 over D. And if X minus D is below your target toxicity band, then you should escalate. If X over D is in the target toxicity band, then you stay. If X over D is above the target band and X minus 1 over D is below the target band, then you stay. Otherwise, you de-escalate. So, and that's it. Those are the I3+3 rules. BOIN is similar with a couple of differences. Rather than being used directly, the upper and lower bounds that you specify for your target band are converted into boundaries to be tested and that minimize the expected decision error. And the final MTD is selected actually after applying isotonic regression across the data on all the doses. But that's only done at the final selection. It's not done during the escalation stage. MTPI is slightly different again. Again, it just looks at the data on the current dose. And again, it divides the probability of toxicity into 3 bands, the below target, the within target, and the above target. And then it does a simple Bayesian beta distribution, and it uses the unit probability mass, probability of being in the interval divided by the interval length, to calculate which is most likely, that you're below the target, in the target, or above the target. And of course, if you're below the target, you escalate. If you're in the target, you stay the same. And if you're above the target, you de-escalate. There is a second version of MTPI, MTPI2, that actually is claimed that for typical values creates better decision rules. We haven't implemented that in facts at the moment, and so I haven't used MTPI2 in the example. And one of the neat things about these methods is that before the trial runs, because you're only considering the data on the current dose, you can create a simple decision table. And that table then becomes all you need to actually execute the trial. So in this decision table, so you can see I've got 3 columns because I'm assuming we're just doing complete cohorts of 3. So if we've tested 3 subjects, 6 subjects, or 9 subjects, and then the number of observed toxicities from that 3, 6, or 9 subjects. And then in the cell, we show the decision to either E, escalate, S, stay where we are, D, de-escalate, or X, de-escalate and ban this dose from the trial, never re-escalate to it. The right, I think the trying to get the order right, which I should have put on, or sort of written up here. So these different decisions correspond to the three different models: the I3+3, the MTPI, and the BOIN. I think that is. Although maybe it's maybe I've got MTPI and BOIN reversed. I think I have, because I think there's an oddity in the BOIN that with cohorts of 3, with the BOIN, there's no decisions to say stay at the same dose until you've got a cohort of 9 with 2 toxicities. But you can see the 3 methods give slightly different decisions in some of the cells, and of course these cells happen to be the most important ones in the design. What you do when you see 1 toxicity out of 3, 1 or 2 toxicities out of 6, or 2 or 3 toxicities out of 9. So in this example, I'm going to, I had, in the fullness of time, I will repeat this with 7 doses. So far, I've only done, and we've certainly only have time today to look at the 11 dose example. So 25 milligrams is our safe dose, de-escalating back down to 37.5 is our starting dose, and then we're going to go up in steps, sort of initially, sort of doubling and going up by about 50%. So 37.5, 50, 75, 100, 150, 200, 300, 400, 600, and 800. Our target DLT range will be in the range 0.15 to 0.3. So if you like, the target toxicity is the midway between that of 0.225. And what I did was to create 10 scenarios where the MTD was at each of the doses in turn, except for the safety dose, mixing steep and shallow gradients, and I used the parametric models in FACS to generate these, so that I wasn't manually entering doses. Only one of these models will actually match the BLRM in FACS. So there's only so not all the scenarios will look linear in the logit space, and so they will, have we're not biasing the scenarios towards ones that kind of naturally get fitted by that model. So there may be additional options that would be interesting to explore, but to keep everything on a kind of a level playing field for all of the different designs, we're going to use a cohort size of 3, a maximum of 15 cohorts, and we're going to stop when we have 3 cohorts on MTD, and we start at the second dose at 37.5 milligrams, reserving 25 milligrams as a fallback dose. And we only increase by 1 dose strength at a time. In the N-CRM, there's a possibility of what's called overdose control, or EWOC, escalation with overdose control, where you can specify this maximum tolerated probability that the toxicity is above the target band. And I've set a rule that I want my maximum tolerated risk of being above the band is 50%. And a second rule that my maximum toleration of the probability is above twice the target band, 0.6, is 5%. So in MTPI, pretty much all we have to enter is that we've got 11 doses. The starting dose is 2, the cohort size is 3, the maximum cohorts is 15, and like the other methods, it's got this maximum probability that the dose exceeds toxicity, and they set a much higher level because of the way they calculate the probability. So here it's set at 0.95. So we'll just look at entering that in FACS. So we start facts, we select the kind of design we want. So we're down here in the dose escalation designs, and we want to do an MTPI. So we double click on that, and we get presented with the tabs to input the parameters for the MTPI with some default starting values in. So I need to change those. I said my maximum cohorts was 15. I said my range was going to be 0.15, the middle of the band being 0.225, and the upper band being 0.3. I was going to have 11 treatment arms. I could go, so the dose index you can't change. I could change the dose names to represent what I'm using. 37.5 milligrams and so on. And then I need to set up the response profiles. Sadly, we don't include the parametric models here in the MTPI, and I will need to create them under the N-CRM and then copy the corresponding toxicities over manually, which is obviously a bit of a pain. And I'm going to turn to one I prepared earlier rather than make you watch me do that. The last tab before we get to run simulations is the design tab. And here it shows the decision table. Here it's showing it for every possible number of subjects on a dose and every possible number of toxicities. We can trim this by saying, well, I'm going to stop when there's 9 subjects on a dose. And here we've got this maximum probability that the target toxicity exceeds 95. And there you can see the escalation, the de-escalation, and the stay at the dose decisions, and the DU, which is de-escalate and do not return choices. If we lower that probability, you'll see we get more DUs to a point where it might actually be very hard to see a trial execute. So it really does have to be at quite a high value to have a workable design. And that's it. That's all you need to enter to do an MTPI trial. And entering a BOIN design is very similar. Maximum number of cohorts, cohort size, bounds, treatment arms, scenarios, and then DU and the maximum number of subjects. So here we have one already entered for the MTPI, and here we've run the simulations, which are very fast because there's no math going on, and we can see the results, and we can see lots of operating characteristics for each scenario, how often each MTD is selected, and then we can look at other things such as probabilities of MTD selection, the mean fitted toxicity at the end compared to the true toxicity, number of escalations and de-escalations, and so on. A good highlights is to look at the distribution of the MTD per scenario. But obviously we've got a number of scenarios and we're not going to look at each one. And we need to look at where these scenarios came from. So I've done a similar thing, but I'm now using the N-CRM. And I'll cover the N-CRM parameters in a minute, but just look for a moment at the where the scenarios came from. I've used these model generated scenarios where I've picked one of the 3 models, the logistic, the Emacs, or the log logistic. And then for each one, I've selected the parameters. Now, for all of these, I've left the minimum and maximum toxicity as ranging between 0 and 1. And then, so the two parameters that get modified are the slope parameter and the ED50. Well, the Emacs doesn't have a slope parameter, it only has an ED50. The logistic, here we've set the slope at 0.05 and the ED50 at about 70. Here using the actual dose strengths of the different doses. So the doses here in the N-CRM, I've actually entered the milligram value as their corresponding dose strength. So if we go down the scenarios, you can see cycling through the different models, variously making relatively flat curves or relatively steep curves, ensuring that one of the doses, and that's the dose named in the profile, lies in the target range. So here 300 lies in the target range. You can see it has a toxicity of 0.2689. All the way out to a relatively steep curve, but with a very late onset of toxicity. So FACS calculates the corresponding toxicities and displays them here, which has then enabled me to manually copy them into the MTPI responses. And similarly, I've copied them into the BOIN and the i3+3 online tools. So just looking at setting this up in FACS, sorry, the N-CRM. So here now, instead of MTPI, we pick N-CRM. We have a number of options here which we're not going to use. So I'm recruiting in cohorts. I'm not including efficacy. Again, I'll set a maximum of 15 cohorts and a maximum cohort size of 3. I've just got one study population. We're not doing cohort expansion. My toxicity, again, like the MTPI, we set the bounds of the toxicity target band, which is 0.15 to 0.3. Don't need to specify the middle. If we don't want to use a toxicity interval, we have got the option of simply saying target a particular toxicity rate. Then we specify the overdose. The default here is a little bit low of 0.25. That's going to, in some cases, possibly make it difficult to escalate. So we're going to have to set it to 0.5. And then, so that's, don't escalate if the orange and red bands total 50% of the posterior probability. And then if the red band itself is above 5%, we'll also prevent escalation. Then again, we need the 11 doses. And as I said, in this example, I actually used the dose, the dose strength as the in milligrams as the dose strength. And you can see I had a little warning sign that when the right dose strengths weren't monotonically increasing. But now I've completed entering them, it's happy and those have gone away. So then in the virtual subject responses, I could add profiles where I manually specify the toxicity, or I create those model ones that I've already shown you. And then we come to the design. So in the N-CRM, the design has got a number of steps. You can see these 5 sub-steps. So it's got a lot more to choose here than in MTPI or BOIN. The first is for the model, what is the reference dose? Where are we going to anchor the dose? Where's the effective dose strength going to be treated as 0? Now I think in this example, Yes, we set it at the first dose. So we said the reference dose is 25. And then we've said the effective dose strength, we're going to use log of the dose ratio of the doses to the reference dose. So here, the dose intervals are considerably less than 1, 2, 3, 4, 5. You can see these x-hat values. We're stepping up in values of about 0.3, 0.4. Then we have to specify the toxicity response. And here we're specifying the prior values for the two parameters for the linear logistic, the alpha parameter, which is the intercept, the value of the model when we're at the dose with a value of 0, an x-hat of 0. So that's at the 25 milligram dose. And then the value of the slope parameter, in this case, the log of beta. Now, a way I like to do this is to use a little tool we have, which allows us to derive a prior. There are a number of prior derivation methods. Now I can't do it here because I've only entered one scenario. But what I like to do is to say, well, derive the prior from the scenarios. Treat the scenarios as a full range of uncertainty in the toxicity that we may need to model, and derive the prior distributions based on those. So it fits the logistic to each one of the scenarios in turn, gets a set of values for alpha and beta, and then says, well, what is the corresponding bivariate normal that matches those sets of pairs of alpha and beta? And that gives us a prior, which if we sample from that prior and fit 100 logistics, it gives us this range of curves here. So you can see that the prior is informative in the sense that we say, well, there's very low expectation of toxicity at the very first dose, but it's pretty uninformative from kind of dose 4 onwards, and it's also pretty uninformative in what the slope is likely to be. We can do things like specifying prior pseudo-subject data, prior data in terms of pseudo-subjects to be added in to the data. And then we can get to these two rules, the allocation and the stopping rule. And here I'm simply saying, don't do a run-in, simply start at dose 2, allocate to cohort, and don't increment by more than one dose step. And then for the stopping rule, simply stop when we have 3 cohorts on the MTD. And then that allows us to get to this point where we run the simulations and get all our operating characteristics. And a graph we should have in MTPI, but don't yet, that is there in the N-CRM, is a sort of summary graph of how the simulations are performed in terms of selection of MTD. So this is a histogram for each scenario of the number of times each dose was selected as MTD. And then it's been colored in because FACS knows the truth of the toxicity rate we were simulating and whether that truth was below, blue, in, green, above, orange, or way above, red, the target toxicity band. So you can see reassuringly that in almost all the scenarios, the peaks of MTD selection are green. Here in dose 4, the dose below the toxicity band is a close second. And here in this simulation, we do go above the toxicity band. It's likely that both these scenarios contain doses with toxicities that just lie outside the toxicity band. And so it's understandable that they get selected a number of times. So the BOIN software was quite good. We could load the scenarios from a file. Sometimes it would time out. I'd have to reload things. There is a desktop app available. It exception didn't do anything. with no explanation, but I was able to deduce this was due to Windows security issues, which I was able to disable. It does provide some view of individual simulations as animations, but only on the Shiny app, not on the desktop. The only allocation it reports is patients as a percentage of the total, which is, it would be nice to see the actual numbers. And also you can't get hold of a full set of simulated individual simulations to do other kinds of post-processing, which of course you can in facts. The I3+3 was way worse. It had no stopping rule. It only summarized for patients allocation for the first 10 doses, and entering scenarios was very, very fiddly. So it's a summary of the scenarios, the individual toxicity rates being simulated, with the ones in red being ones where we're simulating rates that lie inside the target toxicity band. Decision table. This is what plotting all the scenarios looks like on one graph. So you can see we have got quite a full range in terms of where the toxicity is, where the target toxicity is, and what the slope is. And if we plot them in as logits, you can see that they're not all linear. So there are ones which are awkwardly shaped in terms of just fitting a linear logit. So the scenarios aren't biased towards fitting the Bayesian logistic regression. That's what the prior looked like that we saw. So how do we compare these designs? So we've got quite a few operating characteristics. One of the things we want to do is to try to make sure they're all got stopping rules that gives them roughly equal sample sizes. If you have a lot, if one design ends up using a lot more patients, it will of course cost more and take longer. So its results are not really then directly comparable in terms of how well it's done to find the MTD. And annoyingly, the I3+3 code had no early stopping option. I did create then a second N-CRM, which used a much larger sample size to compare it to the I3+3. I added a stopping rule that I also wanted the probability of toxicity at MTB to be at least 60%, as well as having 3 cohorts on the MTD. So I'll come back to the loss function in a minute. So yes, we could see these results in the N-CRM. So we can compare sample sizes. So the first 2 columns are my larger N-CRM and the I3+3. So you can see the large N-CRM, even though it is larger, it's still stopping early, particularly in the scenarios where the target dose is at the low doses. So it's comfortably using fewer subjects than the I3+3, except when we have to go right to the very end of the doses. And then for the other 3 designs, MTPI, BOIN, and my first N-CRM, there you can see that they have roughly similar sample sizes, honors of being the smallest sample size, being kind of shared between MTPI and N-CRM. The BOIN is always a little, interestingly, is always just that little bit bigger by roughly a cohort. And then we can look at what proportion of times the designs pick an MTD in the target band. And interestingly here, so the large N-CRM does better than the I3+3 in every scenario. The I3+3 does better than most of the other designs, but then that's because it's using a much larger sample size, 45 subjects instead of 20 odd. Between the 3 designs using roughly similar sample size, interestingly, the N-CRM largely does the better job of selection of MTD. But I wanted to compare. So the interesting thing is, well, what if you're picking a dose that's not in the MTD? Are you picking a dose that's unsafe above the target band, or are you picking a dose that's just not as effective and below the target band? Are you doing it when And are you picking a dose that's way outside the target band, so the difference in toxicity is quite big, or are you picking is it when you're picking doses that are actually very close to the target band? So I created a loss function. So if you've picked a dose in the target band, you score 0. If you pick a dose above the target band, the loss is 200 times the difference between the true toxicity rate of that dose and the top of the target band. And if you pick a dose below the target band, the loss is 100 times the lower, the difference between the true target rate and the lower bound of the target band. And here the N-CRM is no longer quite such a universal winner. So presumably it's actually tending to stray above the target band a little bit more with this particular design anyway, than the BOIN. MTPI is definitely the worst. So the green values are the ones between these three that have done best, and red is where they've done worst. So it'd be clearly interesting to see how much MTPI2 improves on that. But it was clearly, you can see why MTPI needed another version. It's not got the best decision making. So that's a quick look at using FACS to simulate dose escalation, and a quick way of trying to compare different dose escalation options. So the future is in dose escalation is going to be very strongly driven by this new FDA initiative, the Project Optimus. And there's this growing concern that the identification of the correct dose in the standard oncology development program doesn't do a good enough job. And there's too many drugs being approved at a dose which then needs modifying post-approval. And so the FDA are leading an initiative to promote better trial designs for oncology. So it looks like the implication in phase 1, and this is also partly because of some of the new types of oncology drugs, but in dose escalation, we won't just look at MTD, but we need to look at efficacy as well. So we want to look at the minimal efficacious dose or the optimal biologic dose, that is one that maximizes a sort of benefit risk, or provide a desired therapeutic effect while minimizing toxicity. So somewhat like finding an MED. And there's some specific challenges. And I don't think that there's been a couple of papers of suggested designs. There were some at the ASA Biostats last week, and they're still not meeting all the issues that the FDA are identifying, which include that one of the complications is that the efficacy endpoint is usually observed later than the toxicity endpoint. And that's, yeah, and the problem there is simply it's going to make the simulation a bit more complicated. Plus, you're going to have to make the interesting issue of making decisions. You know, do we make decisions based on the toxicity we've seen, or do we wait for the efficacy? Kind of what do we do while we're waiting for the efficacy? They suggest the efficacy should be modeling or looking at the low-grade toxicity, possibly as well as the DLTs. Obviously, to get around this first problem, people want early efficacy biomarkers, but the FDA aren't convinced that there's many good ones around. And they also say, and this will be a problem, that don't assume efficacy is monotonic. Obviously, and also that the efficacy endpoint may not be dichotomous. And maybe what you should be doing is not picking a dose, but identifying a range of doses to make further dose selection in phase 2 or phase 3. So we can already do a bit of what Project Optimus might be looking for. And so we'll look at that now, but it's certainly not everything. In particular, we've got limitations that that currently we're simulating the efficacy and toxicity endpoints being simultaneous, occurring at the same point. We only have a dichotomous efficacy endpoint model, and it assumes monotonicity. But I think we've already got some interesting capabilities, which I shall hope to show now. So doing toxicity and efficacy just builds on the toxicity model that we've already had a bit of a glimpse of already. So all the features that are in the toxicity model are available when you're looking at toxicity and efficacy. It isn't some kind of cut down, simplified N-CRM. There is a completely separate model for efficacy. Also, as I said, it's also binary and using a simple logistic regression model. And what the design does is it separates the trial into two stages. Pardon me, the first stage is called the find the MTD phase, and it runs just like a conventional dose escalation study. And then when it meets the stopping criteria, that might be the same stopping criteria as a conventional dose escalation, it flips to finding the minimum of efficacious dose. The idea being that it should then be able to track back down to the lower doses where it saw sufficient efficacy. Of course, you're not guaranteed that the minimum efficacious dose is below the MTD, and the rule is still all the overdose protection rules still apply. So if the MED is above the MTD, then you're restricted to allocating no higher than the MTD. And once you reach a sort of a second cap of number of cohorts on that MTD, then the trial will stop. Basically saying, well, we can't search for the MED; it's too high. So all the while doing the second phase and looking for MED, of course, it's still getting toxicity data. It's still updating the toxicity model, which may shift where the MTD is. But as there is this kind of growing sense, well, you don't need to go in the modern dose escalation, you don't actually need to go all the way up to the MTD. It turns out we can already do that, in fact, because there's one of the stopping rules for the first stage, find the MTD, is what's the maximum number of cohorts to allocate in this phase, and you can simply set that to 1. So it allocates the first cohort on the first dose, which is what you're going to do anyway, and then it automatically switches to just hunting for the MED, of course, all the while still estimating the MTD and making sure you don't escalate above it. A feature I haven't shown today is open enrollment, where in N-CRM you don't simply allocate in cohorts, you allocate continuously with a maximum on the number of incomplete subjects, which allows you to enroll somewhat faster. And what you can use is what's called a backfill rule, which means that when you reach the maximum number of subjects who you're still waiting for toxicity information on, subjects who are still available to recruit, you can recruit, but what you do is you don't put them on the current dose, you put them on dose below or a dose below that, with various rules that you can use to restrict how many you do and how far below the current dose you go. And of course, while you're escalating looking for the MTD, this means you're allocating these patients that you couldn't otherwise use onto the doses that you hope to go back down to look for the efficacious dose. And lo and behold, you should find you've already got more data there than you would have if you hadn't been using backfill. So if we look very quickly at I've created a slightly different example. I've only got 7 doses. They're equally spaced. So I've got a smaller number of cohorts, 12. I've stuck to the cohort size of 3. The toxicity band is a little higher, between 0.2 and 0.35. And my overdose control is simply that the probability of toxicity, that the toxicity is about 0.35, shouldn't be greater than 50%. And for efficacy, I'm requiring my target. I'm targeting the minimum dose with a rate of an efficacy rate of 50%. I'll create some scenarios. Those are the numbers. A bit hard to make head or tail of what that's going on, but the clue is in the scenario name. Oops, sorry. So the first table are my toxicity scenarios, and the lower table are my efficacy scenarios. And S2 means that the target dose is dose 2. S4 is the target dose is the 4th dose. In other words, that's the MTD. S6, the MTD is the 6th dose. And my these were hand entered, and the parametric example actually has the MTD at the top, the 7th dose. Conversely, the efficacy scenarios, the target minimum efficacious dose here is respectively dose 3, dose 4, and dose 6. So these are entered separately, and FACS then does a cross combination of all of those. So I end up with 12 scenarios, with each of the efficacy scenarios paired with each of the toxicity scenarios. So you can see I'm going to have scenarios where the toxicity is too early and it's going to prevent me reaching MED, and then scenarios where the MTD is sufficiently high and I will be able to reach the MED. Again, I've derived my prior from the scenarios, but here, because I've got so few scenarios, I then manually tweak them. I've rounded them to the nearest numbers, and I've doubled the standard deviation on beta, and the same for the efficacy scenario. So standard escalation of 1 cohort per dose, don't escalate more than 1 level. Here, I didn't start at the 2nd dose. That might have been a mistake, but they just start at the 1st dose. And then I've got stopping rules for MTD, but these are all effectively null and void, because I've got the rule that the maximum of 1 cohort is used to determine the MTD. And then my rules for the MED is stop when I've got 4 cohorts in the MED. And the probability that I found the MED, or the dose I've picked, has a probability of greater than 40% that it is the MED. If my MED is above the MTD, then keep going until I've got 6 cohorts on the MTD, and then stop. So we'll just take a quick look at what all that looks like. In fact, so entering the study information is very similar to before, except I've now checked I've got efficacy as well box. Toxicity tab looks the same. I just
Tom,
yes.
One question.
Yeah.
Is FACS assuming the efficacy observation window is the same as the toxicity observation window?
I'm afraid currently it is. Yes. Yes.
I assume we are considering changing that.
We are indeed. I've got a little page at the end on what we're expecting to do next year.
All right. Awesome. Thank you.
So again, on the toxicity tab, again, we specify the band and the overdose limit. On the efficacy tab, it's much simpler. It doesn't have bands. We simply say, what's the target MED rate? And do we want to find the dose that's nearest it, highest below it, or lowest above it? We specify our treatment arms again here. I've just left the dose strengths as ranging from 1 to 7, just simple linear. Those, I've created the 3 toxicity profiles I showed, plus a parametric toxicity profile, and then 3 efficacy profiles, which happened to be done parametrically. And then the design, the X-hats, unlike the other design, like the other design, I set the reference dose to the 1st dose, in fact, below the first dose, so dose strength 0. And then I said simply that my transformed dose strength is simply a linear difference. So in fact, the transformed dose strength remains 1, 2, 3, 4, 5, 6, 7. If I derive the prior from the scenarios, get values like alpha, for alpha -5 as the mean and 1.4 as the standard deviation. So I go just -5 and 1.5. And for the beta, I get a derived prior of -0.14 with a standard deviation of 0.22 and a rho of 0.905. I would have probably meant to just change that to be a rho of 0.1, and I've rounded the log beta down to -0.15, and then I've effectively doubled its standard deviation to 0.5. The most important thing in the prior is to make sure you've got the right amount of variability in the expected slope. And then a similar thing for the efficacy, although you can see here there's quite a bit more information in the efficacy prior. As you can see, a type 1 error would be huge. The prior is definitely saying we haven't got a flat efficacy. Maybe we should consider that. And there's also, interestingly, quite a high correlation, negative correlation between the early efficacy and the slope. So the less efficacy we see early on, the higher we expect the slope to be. Again, no use of prior pseudo subjects. Again, a very simple allocation rule. And then the stopping criteria I mentioned, where the crucial rule for the stopping the MTD phase is that stop up to 1 cohort, and then stop the MED when we get 4 cohorts on the MED, or 6 cohorts on the MTD if we can't escalate that high. So we now get some interesting across scenario graphs. So the first one, as before, shows the selection of MTD. And we might be a little alarmed about how high these are in some places. But in fact, what's happening here is this is when the MTD is somewhere above the MED, and we're simply not escalating and allocating doses high enough to properly estimate the MTD. And if we look at the MED, then it's either being shown as being at or above, under or above the true target. I've got no doses which are bang on the target. And of course, we can see it's doing a fairly mixed job. Could be a bit more accurate in this one. But because although it's recording it as being above the MED target, it's obviously the closest one to the MED because the dose below is below the MED target. Perhaps more interesting is what we call the OSD, the optimal selected dose, which is a bit of a posh way of simply saying, if the MED is available, pick the MED. If it's not available, because it appears to be above the MTD, then pick the MTD. But now we have entertainingly colorful graph where we've got, for instance, these ones here, the peak is at the in these pale green, which is target and ineffective, which means that we're selecting the MTD, we're at the target toxicity, but it's ineffective. And that's because these are all scenarios where the MTD is at dose 2 and that's below the MED for any of the efficacy scenarios. In the next 3 scenarios, you can see that the prefix is S4, toxicity is at, the MTD is at dose 4, and we get peaks with this dark green, which is at the target and effective. So we're actually at the MED and effective, or we've got these lighter blue ones where we're actually under and ineffective. Ineffective is a bit strong when we we're actually targeting a specific toxicity rather than an efficacy band. And then in the third one, where the MED is still above the MTD, then again, our peak is at the light green, where we're at the MTD and can't go as high as the effective dose. And then we have a range where now the MTD is at dose 6, and we start to have more under toxic but effective. So the dark blue, except when they're both at dose 6, where we have some under the toxic and ineffective, or a little bit of light green, where we're at the target and effective. So although we think efficacy is dose 6, it's obviously a little bit below. It's probably 0.4 something rather than 0.5. And then lastly, where the MTD is right at the very top dose, again, you can see where the MED is now being selected, and we're not capped by the MTD. We can check on how well we're fitting the toxicity and efficacy, and we can look at the allocation. That's a little hard to read. We can also look at some allocation histories. Not that one. So here, for instance, efficacy is at dose 4, toxicity is at dose 6. So we can see here we've got two histories, one showing the toxicity and the other the efficacy. And we can see here that we saw a couple of toxicities and it stepped down, but it was also seeing sufficient efficacy, so it was happy to stay there. In the second simulation in this scenario, it saw no toxicity at all. So as it was escalating, it started to see efficacy, saw enough at the 4th dose, and then just stayed there. It tried dropping back down, saw no more efficacy, and then selected dose 4, which is actually the true target in this case. And so on. We can look at dose 3 looks remarkably similar, except it saw one toxicity. And dose 4, it saw one and stepped down, but then it saw enough efficacy and was happy. So I think being able to look at the individual simulations is really important for kind of getting a good sense of how the design performs, and also of presenting back to the clinical team, well, these is the decisions this design makes. Given this data, are you happy with it? And you can just review a whole series and make sure they're comfortable with its escalation and de-escalation decisions. Here, the color banding is showing the truth, which of course might bias their decisions, and so we can show them the graph without hidden, and all they get to see is the actual observed toxicity and efficacy. We can also look at graphs that show what the actual model is doing in a scenario, and also look at the estimated probability of being below, within, above, or in unacceptable toxicity as the simulation plays out. So I hope there is then, importantly, there is a tab, we call it the analysis tab, but you can actually enter specific data, run the analysis, and get given the resulting decision rules, as well as the fitted data and the posterior expectation that the probability of toxicity is below, within, or above the target band to actually use as the trial runs. Interestingly, the sample size varied very little between the scenarios. The proportion of toxicity, the proportion of subjects that experience toxicity varies largely, greatly, depending, of course, where the toxicity is on the dose range. And the proportion of efficacy, if the MED is less than the MTD, so the proportion of subjects that experienced efficacy was nearly 50%, was typically in the high 40s. So to conclude, what we plan to do with FACTS next year is, along with some other work in other areas, we want to bring dose escalation up to date. So that means enhancing the MTPI to either be MTPI2 or at least have the option to be MTPI2. To add in a BOIN model and an I3+3. For N-CRM to extend the backfill option to even when you're just doing cohort enrollment, and to be able to simulate times at different times for efficacy and toxicity, and also to allow the escalation to target the MTD of a lower grade toxicity, not necessarily a DLT. In fact, you'll be able to target both MTDs, both the lower grade and a DLT MTD. And those should be, yes.
Quick question here.
Yeah.
So is determination of the optimal dose, is this always a comparison to placebo? My question is, can you set this up to be both on an absolute scale for definition and a relative scale to placebo, or is it only one way or the other?
So you can do both. Everything I've shown you today, I was using in comparison to an absolute. But yes, it is possible to include a placebo dose and compare to it.
Okay.
Most of these enhancements I'm pretty confident about. They're straightforward, with the exception of simulating different times for efficacy and toxicity. Because dose escalation hasn't had to worry too much about time up until now. That actually is a little bit more restructuring of the code than it might look on the surface. But the rest should be fairly straightforward. And then there's just the main references for the different approaches. So any last questions or observations, Kurt?